Okay, so the next step is for clustering visualization and annotating the single-cell RNA-seq clusters. So when we have the single cell, in the early days, people really try to get the readout out of every single cell. But no matter how hard they try, even with SMART-seq, the readout you get from a true single cell is never going to be as accurate. And so people realize that, okay, so if we, in that tissue, you probably have 10 or maybe 20 different cell types. You know, if you think of your brain, you know, you take a slice, there'll be some gray matter, white matter, different, you know, different cell types. In a tumor, there will be the cancer cells, the normal epithelial cells, and then there were some different immune cells. So it's a limited cell number. And so if we figure out these cells belong to the cancer, these cells belong to the, say the white blood or T cells, those belong to the B cells. If you can look at the cell in clusters together, that will give you much more accurate readout. The difficulty here is we are trying to cluster. So, you know, clustering a cell is good, but we're trying to have very high dimensions. In this case, thousands of cells, and each cell has like tens of thousands or 20,000 genes in here. Even though that matrix is very, very sparse, you still end up being, say, 10,000 columns with 20,000 rows, right? So if you want to do clustering, those can be kind of tricky and also very noisy. So we want to do some dimension reduction. If you remember our original dimension reduction lecture from last Thursday, we want to use few dimensions, maybe not quite 2, but you could imagine 20 principal components, or maybe up to 100 to represent the most of the variations in your data. That's already a lot better than having 20,000 genes per cell. So in here, you can see if we run PCA, also you can run PCA on the expression matrix. This can be either done on the count, the exact count matrix, or if you converted your data into TPM, you can do a TPM divided by 10 plus 1, just kind of to make sure that basically if the expression is really low, is as good as 0. So if you detect, and then also having this 1 is to make sure if you have indeed a 0, you can take a log of 0. Okay, so basically this approach will just mostly figure out which genes are roughly expressed and not expressed. The rest doesn't matter so much. You just wanted to help you roughly separate the cell types a bit. And so if you do run the PCA on some single cells, it depends on the experiment. So supposedly, if you are doing a cancer cell line that's growing very, very fast, and let's just do a pilot experiment. We're not even treating the cell with some drugs yet. And you just look at this cancer cell that's growing fast and you do a single cell experiment. You might see that the first principal component, which really separates the noise in the data, is cell cycle, because the cell is growing fast, right? And so when you are doing the single cell the library preparation for the RNA, you are taking a snapshot of the cell at that moment when it went to the machine. And so you might capture some cells at the G1 phase of the cell cycle, other cells at the S phase or the G2 phase or the M phase of the cell cycle. That may not be the most interesting thing you want to even look at, right? So you say, well, let's say I have a cell line, I treat with some IPS for reprogramming the cell into an embryonic stem cell. There's going to be a small population of cells growing, moving to that direction, but the remaining cells are still going cell cycle. The cell cycle part is not what you want to look at. Then you want to, you know, maybe use PCA to remove the cell cycle genes. And computational pipelines for single cell now has that feature. You can at least see it because they know what genes are related to cell cycle. You can see if you separate by that, do you really separate the cells? And they can remove that. You can remove some principal components out of the readout. And the remaining one is the real difference of some drug treatment or tissue difference. So for the remaining top principal component that really captures sufficient variance from the expression matrix, you can then use the PCA. You say, oh, I want to get the top 80% of the variation. So depending on how complex your tissue is, you might use only a few principal components, or people use 20 or like up to 100. That would be sufficient instead of using all 20,000 genes. So with the dimension reduction, we can do clustering of the single cells. So each dot represents one cell, and that's being represented by, say, 20 numbers, the top 20 principal components. Remember, this is a linear combination. So each principal component is a linear combination of the genes that you read in that single cell to capture the variations for that single cell. And then the second principal component is a different linear combination of all the genes in this single cell with a different weight, which is orthogonal to the first one. And with maybe 20 principal components, you can already capture most of the variations in your expression matrix. And so with here, you can imagine we are trying to run a clustering. Each dot have, say, 20 numbers vector, right? There are many possible methods. You can do hierarchical clustering. You can do k-means. You can also use graph-based approach. So currently, this graph-based approach is pretty popular in the single-cell world. So the idea is you can first create a graph structure. For example, based on after PCA, each dot is represented by the top, say, 20 principal components, 20 numbers. Based on that 20 number, you can run the k nearest neighbor to find the other cells that are similar. And if they're similar, you can draw a line between them. And so each cell finds its closest neighbors. And then there are algorithms in the machine learning world. For example, this would look somehow like Facebook network. So people are using the kind of social network world. For example, each of this line you can imagine are your Facebook friends, or they are your cell phone contact list or whatever, right? So you can imagine, of course, with Facebook, the network is much, much bigger. And how do you find communities? You say, there's this one group, there's another group. And so you can imagine if you are this person and you have your own network, so people connected to you as well as their own social groups, and you want to see how they are related to each other. It might be, this is your dorm friends, those are your class, people who are taking classes with you, and these are your sports friends, and so on. So how do you determine those different groups? There are some standard algorithms. We will not go through that much detail. I would say this area is still really active. Every year we might be dealing with a different algorithm. So the idea is, after you have this connected graph, there are some tools that will automatically identify clusters. It's called the modularity optimization techniques that can give you some of these groups. And so when you run these pipelines, one important parameter you can tune is called resolution. Because frankly, with the current single cell, say if you are taking just a blood cell and you do a single cell, you ask people how many different cell types or how many different clusters do I need to have? People actually don't know how many different immune cell types. So if this one immune cell have different expression, is that the same cell, just different expression, or is a different lineage already? Because you look at the T cells, how they differentiate. At some point, they're called the different cell types now. Where do you draw the line? It's unclear, right? So when you do the clustering of some cell population, you have to also decide how many clusters do you want. So the parameter resolution is something you can tune. If you have higher resolution, you can have larger number of clusters. You want to look at more details. Even if things are slightly different, you call them as different clusters. But if you have low resolution, yeah, sure, you just need to have, say, 3 different cell types. That's okay. So you can tune these parameters. And so there is no gold standard. I think people just look at it and decide whether it makes sense or not. Okay. So after you have the clustering, we also want to visualize them. In the dimension reduction lecture, we talked about PCA, we talked about MDS, and so that's kind of a natural way to visualize the single cell data. In the early days, when people do single cell, they also use principal component analysis. But later on, a new machine learning algorithm was developed. It's called the t-SNE, or T-distributed stochastic neighbor embedding. So PCA is kind of a linear combination, a linear transformation. And the way the machine learning world, they kind of convinced people that t-SNE might be better than PCA is using this MinNIST handwritten database. And so this database collected a lot of people's handwriting for, how people write zeros and ones and twos. Of course, the database also keep track. They know which one is a zero, which one is a one, right? Some human read it and say that's a zero, that's a one. And then you run some convolutional neural network or whatever, you can run your machine learning. And if you use PCA to kind of embed this, this is at the end how you can embed things. You can see, a lot of zeros are on the top and ones are on the bottom and fours are in here. You see some reasonable separation, but there are things that kind of overlap. Whereas if you use the t-SNE to display or embed this to display this, you can see the numbers now separate much, much better. And so after this, people say, okay, this is something that we can use for single cell RNA-seq analysis. So you wonder, why don't we use this for clustering or use it for classification? So one problem of this is that t-SNE actually does not learn a function so as to classify new data. So if you learn everything with your training data, when the new data comes, there's not a function you can plug in to kind of show how the new data is projected onto the new old one. And so you can't really use it for classification. And so it's only a way for us to visualize the result of clustering. So t-SNE, it also calculates similarity, but this is based on the T distribution. So you can imagine after we have the cell readout, this matrix, we don't need to worry about this handwriting part. But imagine when we have the gene expression matrix, we can calculate their pairwise similarity, right? This is a correlation-based similarities. Interestingly, so the t-SNE basically is trying to use, in this case, only 2 dimensions to draw the figure, to separate out all the cells. And at every time when you are focused on one cell, it's using physical properties from all the other cells. So basically every cell try to pull it towards the other cells based on their similarity measure. And so it kind of look like a MDS. Remember multi-dimensional scaling. We have a distance matrix, right? We have a pairwise cell distance matrix, and then we try to use a 2-dimensional figure to capture as much as the pairwise distance as possible. The difference between the MDS and the t-SNE is that the similarity calculation is transformed from this T distribution. So initially, you might want to calculate, how much is the correlation, and then if the distance, and you convert the original distance into a T distribution, and so you remember T is it's kind of you subtract and divide it by the standard deviation, and so if 2 cells are really close, their similarity is being read out on the Y in here. Whereas when 2 cells are really far apart, very, very far apart, the distance is really small and you don't really care how far it is anymore. So you focus on the distance between the similar points. So for example, if you have another point that's here, you can see that distance is read out from the Y. There is a significant difference from this blue dot because it's kind of further away. But once you are 5 standard deviation versus 10 standard deviation away, it doesn't really matter. You just know this thing is really far away, right? So that's kind of the difference between t-SNE and MDS is you are trying to capture the pairwise distance, except that the distance is being transformed by this t distribution. And so you try to really see how points that are close by, how they are related to each other. Whereas, from one cluster to another cluster, those other points are so far away, you don't really worry too much about them. Okay, so and so for each point, you calculate its distance to all the other points, and then each is scaled by this t distribution, and then also you sum up them to 1. And then that is kind of the distance matrix. So for example, if you have like a single cell data, say if originally, if you do PCA, you might see something like this. Again, you can see here, the dark blue and the light blue are not as well separated, but if you run t-SNE, it's gonna try to really understand the within cluster relationship a little better, but the between cluster relationship, you just know, oh, this thing is really far away. And so by the way, the distance, it's not necessary. Well, if you look at from PCA, you know that the light blue and dark blue are kind of more close to each other. But in a t-SNE plot, you cannot directly interpret the between cluster distance. It's not necessary that the green is further away from the blue than from the red, okay? Because there the distance really doesn't scale at all. Yeah, so basically this t-SNE is a force-directed graph according to the law of physics. Every point is trying to drag the other point based on that distance. If it is close, it will try to drag it more, but if it's far, it has very small force. And that force is based on this similarity scaled by the t distribution. So the points that are really far away has almost no force, but the force the points that are close by really try to position themselves. Okay, so this is, you can see it's kind of similar to MDS, but it's different on the distance measure because the far away point are being really downweighed a lot. It can help you to visualize the data, but it does not explicitly identify or partition the cells into the cluster. You actually don't need to know the cluster information. Every cell based on the distance will automatically fall in. In this case, it happened to fall into different clusters. The algorithm itself does not know that this belongs to one cluster, this belongs to another cluster. This color is being labeled by the clustering algorithm that you did before already. You're only using the t-SNE to visualize this, okay? So we are not using t-SNE to do clustering. The clustering is done with the previous, either the k-means, or you can use this, whatever, the modularity optimization, Leuvian algorithm, and so on. And so you decide, okay, these belong to different clusters, and you label them different colors. So you can see here, based on that cluster, there are some that look different in the t-SNE, but at least you can roughly see it better now. Okay, so this is computationally quite expensive because basically it's looking at every point. So at every point, it calculates the distance to all the other points. It has to scale up, and every point based on the distance will drag the point a little bit towards them. And then you try another point, you look at all the other points, calculate the distance, you move it a little bit, and you do this iteratively. And so this you really need to do dimension reduction first. You cannot do this on the original read matrix, which has 10,000 genes per cell. We usually only do it on the top, say 50 or 100 principal components. And it still runs quite long time. If you have a very big single cell experiment, this can take a very, very long time. So one property of t-SNE is that the distance for the close by points are important. That relationship is maintained, but the distance for the long, like far away clusters, they are not really to scale. You just know that they're kind of distinct. And so last year, we just talked about t-SNE, but this year, I think more and more people are using a new machine learning algorithm called YMap. There are a lot of properties that are similar to t-SNE, but there are some small differences. The first is you can see This is the number of data points, and this y-axis is the time, so it's computationally more efficient. With t-SNE, you can see if you have more single cells, the computation become much more expensive, whereas UMAP, it's a slightly lower slope, so it's faster. But also, if you look at the dimensions, this is actually very, very important. UMAP, you can even do it on the original data matrix, on the count matrix with 20,000 genes. Although I think people still very often do the dimension reduction, but if you don't do the dimension reduction, it's not death. Whereas with t-SNE, you can see it's like when you have more dimensions, the computational time really increased drastically. Okay, and also the advantage of UMAP is it does learn an embedding function. And so if you have, say, one sample that you already have the embedding and you want to add the new data in there, you can use the same projections on this new data to project the new sample onto your old samples, and you can visualize them together. Also, the advantage of this is it gives the same result if you run the algorithm multiple times, whereas t-SNE, every time you run is different, and also every time you add a new data is different, so it could be kind of tricky. And so I think for single cell, a lot of people use t-SNE, but more and more people are now using UMAP for visualization now. So if you look at the results of UMAP versus t-SNE, there are some, you can see these disjoint clusters. It also separates data pretty well. This is from the identical data. So you can see the UMAPs still look quite different. And, we could, at least this is the paper that showed that UMAP is better than t-SNE, is that you can see the same cell types are kind of more close to each other, or are separating slightly better than the t-SNE. Again, the color is not automatically determined by UMAP. UMAP just give you black. You have to specify this cell, I want a blue, that cell, I want green, that cell, I want purple. And that decision is coming from either user specified or from clustering result. So you tell UMAP how to color the dots. Okay. Yeah, so what do we use these kind of UMAP or t-SNE to do? The first thing is you just kind of want to see, how roughly the data look in this tissue, this complex tissue you are looking at. You might want to annotate the clusters. So even though UMAP is not a clustering algorithm, it's mostly a visualization algorithm. You can still roughly see, if you use clustering, you can see, okay, UMAP and cluster does kind of agree somewhat. And then you can look at, for example, this cluster. You can say, okay, let's just look at all these cells. What are these? And then let's look at these cells. What are these? So again, so this part you can see in your clustering, if you use different resolution, this right now is being called as one cell type, but if you maybe have higher resolution, this will be separated into two separate clusters, right? Because within CD4, there are other things like Tregs and Th17, Th1, and things like that, right? So you can separate them further, and you can see these two green things. If you have higher resolution, they might be separated. And so you can visualize this, and then you can use some marker genes. The marker gene can be a single gene. You can also ask t-SNE to draw this color by the expression level of just a single gene in all of these cells. For example, if we look at this one gene, KDR, and this is one, I actually don't even know whether it's t-SNE or UMAP, it doesn't matter. You look at this graph, and you in each cell, you ask t-SNE to draw the expression level of this one gene. And you can see here, this gene is mostly expressed in this one cluster. It's not always there. You can see a lot of those gray dots there. The reason it's not always there is because of dropout. In some cells, this gene is probably expressed, but then you just didn't detect it. Yeah, so whereas CD68, okay, this is it's a well-known macrophage gene. It's highly expressed in here, but lowly expressed in other ones. This can be displayed as a single gene. you can also use some signatures. You give it a signature and you evaluate how much this signature is expressed in one cell or the other, and you can display that. And so remember with the gene set enrichment analysis, previously there is a molecular signature database. There are marker genes already determined as the genes that are uniquely highly expressed in, say, CD4 T cells, macrophage, or dendritic cells. So you could use those gene sets to help you annotate the single cell clusters. This is still really an open question. If you have how to do this automatically, there are some tools that provide some solutions, but this really depends on what signatures you use, what method you use, how accurate it is. So it's, I would say, not as solved yet. I think depends on the tissue. Some you can do better than others if you have seen enough data like that. So one thing you could use is to look at the cell population change between the different samples. Supposedly, I have two samples that I do the visualization together. And this is the YMap figure of everything together. And you can see this, one cell type is 31% of all the cells. Another cell type is a cluster that has 12.5, and another cluster is 9.3. But then you can also choose to say, now let's draw them in one sample versus the other sample. Supposedly one sample is without treatment or control. The other sample is you treat this tissue with some drug. And you can look at them now as, basically you use the same YMap projection, but separate the cells that are coming from the first sample versus the cell that's coming from the second sample that are treated. You might notice, for example, oh, this cell type is now all dead, or maybe it's the expression of this one cell type is shifting, right? So that you can see based on the population of cells that are different between the two samples. You can also visualize the level of gene expression in those different cell clusters, and those can also help you with identify the cell clusters. Okay, so there are also algorithms to say, okay, this group of cell, it has a unique, sometimes you can use it to identify a new single cell cluster. It's a new cell type that people didn't know before. So imagine, supposedly this is your CD4 cell. Previously, people think it's all CD4, but if you look at a resolution, you say, I think this is a new cluster that people never know is some intermediate stage between this cell to the next stage, right? You can identify that, and from there, you can identify some important cell markers. Then you can use flow cytometry to just sort by that cell marker to understand that cell type better later. Okay, so that's kind of some of the ways to visualize the expression in the different clusters. Questions on this? Yes? What makes the YMap algorithm? So the question is, what is the difference between YMap and the t-SNE? I would say the iterative learning process is similar. They calculate the distance also slightly different. To be honest, I don't know all the details. I read some things online, but I can't tell you that I know all of this. A lot of this, I just search online for other slides for what they mentioned, and also watch YouTube videos. So as I mentioned, this is an area that's moving so fast, and every year it's different. And I'm just learning it like a few hours before you guys. But YMap, it has been around since maybe 2 years ago. But I think it's a different. I think it will a lot more math that we don't probably want to go to. Okay, other questions? Okay.
If you cluster your expression matrix in a specific way and feed that twice to t-SNE, what is kind of the step that would make the cluster slightly differently the second time?
So if I I can guess what is why t-SNE give you different results. I think depends on which point you start first to calculate the distance, because the A to B distance and the B to A distance is not symmetric, because each cell, the distance is scaled against all the distance to other cells and sum up to 1. Depending on what is in their neighbor, the A to B distance and the B to A distance is not symmetric. And so depends on which cell you start, you will end up with a different result. And so in t-SNE, there's a seeding process. There's a random seed. Then you end up with a different result. YMap, actually, I don't know how they implement this. They do give you, because there's a projection. Every cell come in, you just project them in a different way, and then you get this result. Yeah, so yeah, that's visualization. So next